Well, we're back once again, and we're going to continue the story of central dogma. And in this case, the big player that we've got is right up here, is of course RNA polymerase. And we're going to be talking about transcription. We'll edge slightly into a little bit about translation and just the overall process of creating proteins from DNA. Critically, we want to revisit in slightly more complex form central dogma. And notice that we talk about this. This is really a nice graph to put it together in two different axes, talking about generation to generation transfer of information of DNA, because DNA replicates itself to make daughter strands of DNA. So we see basically the crossing of genetic information into the next generation, but at the same time showing another one of Crick's observations that not only is DNA the molecule of inheritance, but it also controls the basic blueprint of all cellular processes, functions, organization of the organism itself, and ultimately anything that the organism does. So it's the control of both inheritance but also the phenotype itself. And you can see now we're starting to fill in where central dogma, whereas we did talk a little bit before about replication, transcription, and translation. Notice that we've also included a route here for reverse transcription, that is RNA-dependent DNA polymerase, which can take an RNA molecule and turn it into DNA. We'll talk more about the role that that has either toward the end of this week or early next. Transcription and translation, just as we discovered in talking about replication, was initially identified in the molecular mechanisms shown in prokaryotes. And in this case, prokaryotic transcription is really pretty easy. The DNA contains code for regulation of transcription, and in this case, the initial regulation is the transcription initiation site. Then, of course, there's the particular site in the DNA sequence that regulates translation, and so we call this the start codon. This is where protein synthesis begins and is indicated by the RNA sequence AUG. Then, of course, the coding region that actually codes for the protein or peptide involved. And finally, there's a termination site where transcription terminates itself. So the mRNA codons are translated into amino acid sequence specifically by the ribosome. So you can see it's a very simple two-step process. Of course, here's another great graphic showing you how much more complicated eukaryotic gene expression is than prokaryotic gene expression. You can see that we have the similar idea of the regulation of transcription and then translation existing in the DNA sequence itself. The regulation of translation is identified by the same start codon, or AUG, but notice that the coding sequence of the particular peptide or protein in the DNA is divided up into exons and introns. Exons being expressed regions, however, sometimes they're not entirely expressed, and introns being the intervening regions. So it helps to remember which is which, because exons are generally expressed and introns are the intervening non-expressed regions. There can be many of these in any particular gene sequence. There are, of course, also signals for termination of transcription or transcription termination site. The primary transcript is created. the introns are removed and sometimes called splicing, and then the final messenger RNA is created. All of this takes place in the purple area here, which is inside the nucleus. Once the mRNA is completed, it is transported to the cytoplasm, where, in conjunction with the ribosomes, messenger RNA is translated into the cytoplasm. So we'll see a little bit more of this as we go on. Notably, there are 5 types of RNA molecules: the messenger RNAs we've just discussed, the transfer RNAs, which are the adapters between the amino acids and the codons in messenger RNA. We'll talk about those next mini lecture. Ribosomal RNAs, small nuclear RNAs, which have a role in gene regulation, and microRNAs, sometimes called microinterfering RNAs. It's a good way to remember it, which are short, single-stranded RNAs that block expression of other messenger RNAs. Couple key ideas. Remember that central dogma dictates that genetic information goes from DNA to DNA during replication, DNA to RNA in transcription, and RNA to protein in translation. And what this means is, because we're talking about the flow of information, these are all semiotic molecules. Semiotic. This is the process by which signs and symbols can be used to create meaning. Some of you may have heard of semiotics, very often used to describe code breakers in the military or just on the back of a cereal box stop. Transcription itself involves the synthesis of the RNA transcript, and that's what we'll be talking about this time around. And of course, we will get to the idea of translation. I'm trying to keep the ideas, they are so closely integrated, transcription and translation. We're going to shade a little bit into translation, but really save a little more discussion for that later. If we take a peek at the basic setup for transcription, notice we do have DNA is double-stranded. And in the case during RNA synthesis, which of the 2 strands are actually used to create the messenger RNA? Now, in reality, either of the 2 strands can be used for coding any particular gene. However, the one that is being used at that time is called the template strand, and the other is the non-template strand. So you see in the little breakout box here, you see we've got the 2 antiparallel strands of DNA. One is called the template and one is called the non-template. Notice we always label our DNA and RNA with which end is the 3' and the 5' end, so we can see that they are antiparallel in the double-stranded DNA. Notice that the template strand is indicated in the 3' to 5' direction, and that is because RNA polymerase, unlike DNA polymerase, RNA polymerase moves in the 3' to 5' direction. I'm sure you all remember that DNA polymerase moves in a 5' to 3' direction during replication. This is not the case with RNA polymerase. RNA polymerase moves along the template strand in a 3' to 5' direction, creating a sense RNA strand, that is, a strand of RNA that will be used as a message by the ribosomes to create a peptide or a protein. If you take a peek at the sequences between the non-template and the sense strand, notice that they are actually identical, with the exception, of course, of the thymidine being replaced by uridine. So this is a this particular slide is a little bit confusing for students facing it the first time. The template is the actual template that's used by RNA polymerase to create the sense RNA strand. Just by nature of the complementarity of double-stranded DNA, it turns out that the non-template and the sense strand have the same basic sequence. RNA polymerase functions in very, very much the same way as we saw replication. If you want to express a gene, you've got to unwind the double-stranded DNA. So RNA polymerase exists in what's known as a transcription bubble. It does have some gyrase activity, unwinding the double-stranded DNA, riding along the template strand and creating a sense strand that is similar in sequence to the non-template strand. So what are we thinking about general features? It's similar to DNA synthesis. Obviously, we're going to create RNA, which has ribose as a basic sugar. All of the precursors are ribonucleoside triphosphates. Only one strand of DNA is used as a template at any one time. However, both can contain coded information. And of course, the RNA chains are initiated de novo with no primer. Okay? Here again, and I cannot emphasize this portion enough, the RNA molecule will be complementary to the DNA template strand, but identical to the non-template or sense strand. So again, this is something you have to spend a little bit of time looking at, and the textbook also has some very good illustrations of this. In eukaryotes, things are a little bit different. Genes are present in the nucleus, which of course does not exist in prokaryotes, and polypeptides or proteins are synthesized in the cytoplasm. Messenger RNA molecules function as intermediaries, carried the information from DNA to the ribosomes. RNA synthesis is driven by RNA polymerases and is very, very similar to DNA synthesis. Now, let's take a peek at prokaryotic transcription, and we see we've got RNA polymerase, hits the transcription initiation site, begins to unwind the RNA polymerase, moves in a 3' to 5' direction, which means that the growing RNA chain is in the 5' to 3' direction. So again, remember, of course, the direction in which chain is which as we move along. Finally, RNA chain termination exists when the polymerase hits the transcription termination site. Sorry about that. In general, and this is something we'll see a little bit later on, but I wanted to introduce the idea now, is when we look at a transcription unit, we can number the individual nucleotides, 1 through whatever the end of the gene is, and we always begin the transcription initiation site as being +1. Any bases that occur before the initiation site are known as upstream sequences, and any that follow the initiation site are known as downstream sequences. So again, we're just trying to orient ourselves along the DNA molecule. And this becomes very important when we talk next week about bioinformatics and how we can use computers to actually analyze these sequences. Here's what the promoter actually begins to look like. Notice we have right here the initiation sequence. This is an E. coli, of course. Here we see that the bubble begins, and notice right in here, TATA. This is known as a Tata box, and we see this very typical sequence beginning. Transcription starts then right across here. Notice the non-template strand. Why does it begin with A? Because it's usually AUG. So we've got upstream, downstream. And the transcription bubble moves right along with the RNA polymerase, creating the messenger RNA strand. So what stages do we have? Important to remember: initiation, elongation, termination. Okay? The covalent expression of the RNA chains occur within the locally unwound segments of DNA. That is, that the transcription bubbles are actually much smaller than the replication bubbles. And also, elongation of the RNA stops when polymerase encounters a transcription termination signal. All of this occurs nearly simultaneously in prokaryotes. How about eukaryotes? As you may have guessed, eukaryotes are much more complicated. 5 different enzymes catalyze transcription in eukaryotes, and the transcripts themselves undergo modification, including the excision of non-coding sequences, which are called introns, right? And then some of the transcripts are even modified further post-transcriptionally by RNA editing. We won't get very deep into this idea, but I wanted to keep it in the back of your mind that there are many modifications of the messenger RNA transcript that can occur along the way in the cytoplasm of the eukaryotic cell. Here you kind of gives you the cellular orientation. Transcription, of course, is occurring predominantly in the nucleus of eukaryotic cells. We see the DNA, the transcription bubble, creating the messenger RNA. Note the green and the brown here with introns and exons. And, of course, the 5 different types of RNAs that can be synthesized within the nucleus, transported through the nuclear envelope via the nuclear pores. All of these components enter the cytoplasm where translation occurs, and we'll talk a little bit more about translation in the next mini lecture, so we're actually going to see this slide again. Ultimately, when we think about what happens in eukaryotic transcription, we see we've got the DNA. Here is the exons, introns, exons again. This becomes a pre-mRNA molecule, sometimes called a pre-pro or a precursor. This has a 5' methylguanosine cap on it and a polyadenylated tail, 3' all the way to polyadenylated. And notice that by step 3, the intron is spliced out, and once it's spliced out, we remain with the mature messenger RNA with the 5' cap and the 3' poly A tail. These modifications are very important for regulation of gene expression, which we're not going to go into very much. Just to give you an idea, eukaryotes have 5 different RNA polymerases. And they all have a variety of different functions right here. Here's what the promoter looks like, just to give you an idea. Unlike the simple Tata box in the prokaryotic initiation sequence, and we do have a Tata box that exists in eukaryotic cells, notice that we also have a variety of other promoters, the octamer GC, CAAT, the GC box, another one. And then, of course, here's our transcription start point, which, remember, always starts at 1, so it doesn't start at 0. I love this picture because it shows you the basic three-dimensional structure, even though it's a bit of a cartoon, of RNA polymerase. And notice it looks almost like a hand and a glove, just zooming its way down the DNA molecule. Here is the double-stranded helical DNA, which gets unwound into a small bubble containing two single-strand portions of the DNA, and the beginning, of course, of the RNA transcript. And there is a separate area here, which is where the ribonucleoside triphosphates enter into the enzymes so that the RNA transcript can be synthesized. Really very, very clever. Again, talking about our key points, 3 to 5 different polymerases are present in eukaryotes. Each one functions on a distinct set of genes. We've got the addition of that guanosine cap to the 5' terminus, the addition of poly A tails to the 3' end, and of course the excision of non-coding intron sequences. Remember that some of the information in that pre-messenger RNA, precursor RNA, can be altered by RNA editing, and sometimes this can result in the differential splicing of different exons. So instead of exons 1, 2, 3 being spliced together into a single protein, sometimes they can be expressed in a different, or they can be synthesized rather in a different order, and sometimes only a few of the exons can be expressed instead of all of them at the same time, very dependent upon tissue type and the cellular milieu that exists. And this is just a summary to remind you, these eukaryotic genes contain introns, okay? Introns are excised prior to transport into the cytoplasm. Again, a little bit more detail, mostly reminding you here that introns are variable in size and number. There is no specific number that there must be, and some of them are actually very large, surprisingly large. Again, most but not all of eukaryotic genes are split up into introns and exons. Some eukaryotic genes have no introns. It depends on the size of the protein and also the class of genes that we're talking about. Some of the introns can be very, very large. Alternatively, some of them can be very small, and these form these small interfering RNAs that are involved in regulation of gene expression. People always wonder, what is an intron? Okay. When I was in grad school, we used to call it junk DNA because nobody knew what it was for. Why would you have an intron? Well, that was before the regulatory nature of some of these small interfering and the snRNAs were identified. So there are other kinds of functions that RNA can have, and also, or rather these particular introns can have, and not the least of which could be also structural in nature and helping the RNA polymerase be guided around in these genes that can be very, very large and produce very, very large proteins as compared to those produced by prokaryotes. So really, this is all the information that I wanted to cover today. We zoomed through a little bit of it. Most of the information is here. Please remember to review the slide sets that exist in Piazza under Transcription. And also, please remember to read the chapter in the textbook on transcription as well. You guys take care. Stay safe.